Okay, so good morning or good afternoon or good evening, depending where you are. We have people from all over the place. Um, so welcome to the February virtual Melastomataceae seminar. And today is my pleasure to introduce Luo Chen from the Botanical Garden in Munich and Ludwig Maximilian University in Munich. So Luo is originally from China, and where he did a master's, and he was looking at Podocarpaceae in those old days, and he realized that things without cool flowers, and even though podocarps have very interesting seed and fruit situations, melastomes are way cooler. And he then moved to uh, Munich, where working on the Gudrun Catarite, he started working on all world melastomes, mostly on in Madagascar, but also continental Africa. And I think you will see a very interesting arch of work starting with different techniques and different groups as his dissertation progress. And this is a timely seminar because Luo will be defending his PhD in 9 days. So most of what you see here will be on his defense, but not all, and not everything you see here will be on his defense. So, Luo, it's a pleasure, and please take it away.
Okay, can you hear me?
Yes, all good.
Yeah, thank you very much for your nice introduction, Tobian. Then let's get started. So the title is Advances in the Phylogenomics of Old World Melastomataceae. When Agnes was asking me last autumn, I thought I would be done by this time, but I wasn't sure what to really present. So I thought, okay, maybe I should have a broad topic, broad title to include everything. And then I forgot to change the title. So this sounds really inclusive, and I hope I will have enough to really cover this title. So I think there's no need for me to introduce the family, the audience here. But still, we need to know that the family includes 173 genera and almost 6,000 species now. And most of, not most, but like 60% of them are found in the New World with 84 genera, but also more genera and also more than 2,000 species are in the Old World. Southeast Asia, Madagascar, and Central Africa are the hotspots in the Old World in terms of species richness of Melastomataceae. And in the Old World, we have all three subfamilies of Melastomataceae. So it's Olispioideae, Kibacioideae with only one genus, and also 6 tribes from Melastomatoideae. And the molecular systematics of Melastomataceae started in the 80s when Marty and Dodd sequenced RBCS for Metalis, but they sampled 2 Melastomataceae, and this was the first time when Melastomataceae was actually sequenced, although they only sampled 2. And 10 years later, Conti et al. Also performed analysis on Metalis, and this time they have 5 Melastomataceae. In 2001, Klaus and Rennes and Gudrun and Susan Renner, for the first time, they performed family-wide analysis on the phylogeny of Melastomataceae. So they sampled most of the tribes recognized at that time. And based on this first phylogeny, they also provided some novel insights, like the Old World and New World Melastomataceae, they're non-monophyletic. Also, some tribes were recovered, some were not. They also mapped some key morphological characters to the phylogeny, like the identification of pedoconductive clay. And one milestone study using Asanga data was published 3 years ago by Darren and colleagues from the Melastomataceae book. So they used 2 nuclear and 7 plastid loci, and they sampled more than 2,000 species, including almost 3,000 terminals. and covering most of the genera. So based on this, they built this phylogeny. But we're already in an era where when NGS data has been widely used within Melastomataceae, the sequencing strategies we've been used can be seen here. So we have some RNA-seq data, but not many. I think even as today, less than 20 transcriptomes have been sequenced from Melastomataceae. And we've been doing target enrichment. I will introduce this later. Also genome skimming. Some of us have been doing, and deep genome skimming is basically genome skimming, but with higher sequencing depth. We've been also doing HypeSeq. HypeSeq here refers to the combination of genome skimming and target enrichment. So we have the libraries. Some are enriched, so it's hybridized with the baits. Some are not, and then they're used for sequencing. And since there hasn't been a lot of studies of phylogenomic analysis for Melastomataceae, I can just present them here. In 2016, and Reginato et al., they sampled 16 neotropical Melastomataceae. They sequenced they did genome skimming and studied the plastomes. And 2 years ago, Major et al., they studied a Caribbean clade of Meconia with skimming data. And the first paleotropical phylogenomic study was in 2019 on Sonorili. And we've been also using target enrichment. This started with the angiosperm 353, the universal probe set used by botanists. In 2021, this Metalis paper, they sampled almost 200 accessions of Melastomataceae. This was the first family-wide sampling for the family in terms of phylogenomic study. And this serves as a very valuable resource for us. We also, not we, but the community has also developed our own probe set by Janssen et al. In 2020. And the probe set comprises 384 loci. 60% of them are from angiosperm 353. There are also like 104 loci newly identified by Microminer. There are also 8 single copy nuclear loci that were previously published and included here. Also 6 loci of functional significance. So the fact that we have 266 loci in the Melastomataceae probe set means that we can also have the potential to include those previously sequenced data in the future in our own data set. And this process has been applied to Mammocylon here and the Old World lineage, also on Tibochina. At the same time, our colleagues from China, they've been doing deep genome skimming. As I said before, it's basically genome skimming, but with more data. So this is their phylogeny on Asian Sonorili. The way they did the analysis basically was that they first identified single copy orthologs with newly generated transcriptomic data. Then they mapped all these orthologs to the draft genome of Tigrodioparma is one species in Sonorili. And they only keep those with single copy number. Then they also recover, because from transcriptomic data, you only have the exons. So they also recover the intron sequences from deep genome skimming of one species, Bridia hirsuta. So they have the complete sequence of one gene. Then they mapped a lot of newly sequenced data to this partial genome. This is their strategy. And I think it was last month, they published another paper using the same method. They dived into one lineage of Sonorili called CPS clade, and they described new genera based on the new phylogenetic results. So during my PhD, the aims was to first improve our understanding of the systematics and evolution of the Old World Melastomataceae, In the meantime, we want you to explore optimal approaches for integrating different sequencing data. This includes both Sanger and angiosperm 353 and also different NGS data. So we selected 4 groups to work on. It was Sonorili, Melastomataceae, Dislocated, and Phylogenomics. So first was Dislocated. We focused on one African species, Medinella mirabilis, which is endemic to Central Africa. It's a liana that you find in the tropical forest. The upper leaves are arranged in whorls and the lower leaves are alternate, and the inflorescence is very impressive. The numerous cymes forming a spherical inflorescence. The species was described in 1898, 1 year after the genus was described. And there was only 1 species in the genus. And in 1977, Jacques Felix transferred this genus into Medinella because Medinella is a very diverse, widespread species. So he thought, okay, this fits into the diverse genus. But this treatment has never been really tested in a phylogenetic context. As I said before, the 194 Melastomataceae accession sequencing in the Metallis paper is a very valuable resource for us because it sampled most of the tribes within Melastomataceae. So I want you to first test the phylogenetic placement of Medinella mirabilis with technetium data. So we used all this publicly available data. We also newly sequenced 1 accession of Medinella mirabilis. We performed both concatenation and coalescence analysis, and we found that Medinella mirabilis is not closely related to other Medinella, which is in Sonorili. Instead, it's sister to Dislocated, based on what we have here. But the problem is Dislocated is a tribe with 6 genera, and here we only sampled 2 genera. So we were not sure if it's really sister to the whole tribe or it's actually nested within the tribe. Then we thought of the Sanger data, because in GenBank, there's a lot of publicly available sequences of Dislocated. So we retrieved 152 taxa from GenBank. This covers all genera of Dislocated and more than half of the species. And we newly sequenced 4 accessions of Medinella mirabilis. We used 2 nuclei and 4 chloroplast loci. So this is what we found. It's, again, not really sister to other Medinella, and it's sister to the whole tribe of Dislocated. If you zoom in, we see from here. And we also checked the wood anatomy of Medinella mirabilis and compare it to Sonorili and Dislocated, because usually Sonorili has this scalariform intervessel pits, and Dislocated has alternate intervessel pits, so like these dots, and this is like a whole line. And we found that Medinella mirabilis has similar wood anatomical morphology with Dislocated instead of Sonorili. So we decided to reinstate the genus Myrianthemum based on different lines of evidence. In the meantime, because of the phylogenetic result we have, so the species, the genus is sister to Dislocated. Also, it has alternate intervessel pits. It's a climber. Most Dislocated are climbers. It's tetramerous. The tribe is tetramerous. Also, it has interpetiolar outgrowths, like most Dislocated. Also, they all have berry fruits. So, because of all this evidence, we decided to treat the genus within tribe Dislocated, so that the tribe that was Asian endemic now also includes an African genus. Yeah, to summarize this project, we transferred Medinella mirabilis from Sonorili to Myrianthemum, and we treated it now in Dislocated. We also showed that by combining angiosperms 353 and Sanger data, we could, using angiosperm 353, provide more sequence information, and with using Sanger data to increase the taxon sampling. So first, African Melastomataceae, the tribe is widespread. You can find it in the both Old World and New World. It has almost 800 species in 44 genera. There are almost 200 species in Afrotropics and 130 species from 2 genera in Asia. It was Aspechia and Melastoma. So for the African Melastomataceae, Veranzo Libella established the first classification for them based on Sanger data. As you see here, a lot of the genera now accepted used to be Desertis. So there was a lot of movements within the lineage. But in the meantime, some enigmatic species that couldn't be included before because of the inaccessibility. One of them is Desertis leonensis. It was in one of the it was one of the subgenus within Desertis, but it was never sampled. Also, Desertis splendens and this Dionychastrum schleppanii. So all these 3 species, we couldn't include them previously because of lacking of material. But recently, our collaborators have acquired material of them from recent expeditions. So in this study, we wanted to test first the previously established phylogenetic relationships are still valid with phylogenomic data. Also, we wanted to see where these 3 species will be placed. So we sampled all Old World Melastomataceae genera, including these 3 enigmatic species. It's noteworthy that Dionychia schleppanii was sampled from an isotype specimen that was collected from almost 100 years ago. So in this project, we used the Melastomataceae probe set. We also checked the seed morphology. So this is the phylogenetic result. So despite some differences, clades that were identified by Sanger data before are reaffirmed, including the 2 primary clades, Euheterotis and Pseudoheterotis, and most genera. And age, so within the Old World Melastomataceae, Asian endemic genera and Malagasy endemic genera, they both form a clade and they're sister to each other. And the 3 species I mentioned, yeah, but there are also some genera that were recovered as non-monophyletic, like Melastomastrum here. It forms 3 different clades. One of them is sister to Tristema, so it's a paraphyletic. Also, the Asian clade Aspechia is non-monophyletic with Melastoma nested within it. Also, the Malagasy genera, these 3 genera, they're like none of them is monophyletic. And the 3 newly sampled species, they've been placed. So Desertis leonensis is sister to all the other Pseudoheterotis, and the Desertis splendens is also placed in this place. According to the seed morphology, they are consistent with other Melastomataceae species. So based on this, also the morphology, we decided to reinstate the genus Allodisortis and to describe these two new genera, Allodisortis and Phylogenomics. So this paper has been accepted. We also worked on fleshy fruity sonorili. So sonorili is the second largest tribe within the family. It's also pantropical. There are some in South America, but only in 6 genera and around 13 species. Most of them are in Southeast Asia. So most genera within the tribe have dry capsular fruits, as you see here and here. But there are also some genera having fleshy fruits, which are found in 6 genera here. In the past, they were also called Medinilla Alliance because we were not sure if Medinilla was monophyletic or not, and it was the most species abundant genus. And so in this study, we wanted to test whether or not the fleshy fruities really would form one clade. Also, we wanted to know if the very diverse genus Medinilla will be monophyletic. So similarly, we also used a Melastomataceae probe set. We also extracted plastiloci from off-target rates, and we also sampled all fleshy fruited genera of Sonorili. And this time we had particular focus on Medinilla. We covered most of its biogeographic realms. So we found that both fleshy fruited Sonorili and Medinilla are not monophyletic. The fleshy fruited Sonorili consists of 3 distinct clades. So Chondrichia, and Catentera and Heteroblema, and Medinilla. So if we zoom in, we see here Chondrichia is sister to the whole Afrotropical clade. It's a Sri Lankan endemic. In the past, when in the last paper when it was included, it was recovered as sister to Catentera and Heteroblema. So this is not consistent with the phylogenomic results. And the Catentera, for these 2 genera, they form a clade sister to some Philogathis and Dreissenia species. I mean, in the nuclear tree, they are both monophyletic, but if you check the plastid tree, Heteroblema is actually biphyletic. The Vietnamese species are sister to Catentera, but the Bornean species are sister to one Philogathis species. We didn't really test the possible cause of this, but could probably be hybridization. And within Medinilla, we identified 15 alliances, and we redefined the genus. Now we included Pachysantria and the Plathyandra within the genus. And this work was done by me and my collaborator Peter. So Peter is more focused on the Asian ones, and I was focusing more on the Afrotropical clade. So within this clade, we found that now there are only 2 African species, and they are nested within the Malagasy clade. We also find this westward movement within Medinilla. So like here, this Griff, this Himalayan species, then Sri Lanka to Madagascar, then some dispersed into Africa. Yeah, this paper has been published. And now I'm going to present some unpublished results. So first, we worked on the phylogeny of Sonorili. This was a work published 2 years ago by Lorenzo Lipola et al. I was one of the co-authors, so we reconstructed the phylogeny of African Sonorili. In this study, we also included many Asian Sonorili as outgroups. Although some of the data was working fine with Afrotropical species, but it was terrible in terms of Asian genera. So we wanted to resolve this with NGS data. And for us, what's available, we sampled most of the genera of Sonorili. This includes half of the neotropical genera and all 9 afrotropical endemic genera. We covered most of all but one lineages, Asian lineages identified by our collaborators. We also included 5 sessions of from Melastomataceae as our group, and we have different kinds of NGS data here. We have whole genome sequencing data, we have RNA-seq data, we also have target enrichment data with angiosperm 353, and HiveSeq data that includes target enrichment data sequenced with the Melastomataceae probe set, and also we have deep genome skimming data. So with all these different heterogeneous data set, is there a way to integrate them? So we first tried with the Melastomataceae probe set as reference. As we see here, many nodes are fully supported, and some taxa, they're not really recovered, like Sonorilla appears here and here. Also, the Bornean oxysporoclade is not monophyletic here. Then we decided to develop a new reference for Sonorilla. We included 16 transcriptomes and 2 genome assemblies. This includes 5 from Melastomataceae, 12 from Sonorilla, and 1 Meconia from Meconiae. So we trimmed the reads of all this raw data and de novo assembled the transcriptomes. We identified the coding sequences, and together with the genome assemblies, we did all by all blast. Then we used MCL to cluster them. And for all these possible homologs, we did alignment and inferred single gene trees. In the end, we inferred orthologs. So this is the result. We had over 12,000 homologous genes, and after orthology inference, we had 5,626 orthologous genes left. We mapped all these loci against the angiosperm 353, and we found 176 loci overlapping. And we also blast these orthologous genes against Melastomataceae probes and found 221 loci overlapping, indicating the possibility of integrating different data set. So before showing the phylogeny, I would like to show you the number of orthologs, the difference on this. So, okay, sorry, I think it's wrong here, but this is the result from Melastomataceae probe set. We had on average only 92 loci, and for angiosperm 353 was the worst case. Sometimes we had even less than 50 loci. But with the new reference, we had a way higher loci coverage. On average, we had more than 2,500 loci. And if you look closely here, we can actually see that deep genome skimming performs quite well. So this is the resulting phylogeny. We see most nodes are now fully supported, and all 33 Asian lineages sampled now are recovered. Also, because this time we newly included many African species, and the relationships of the African clades are mostly consistent with the Sanger-based phylogeny. This is not really part of my PhD, but I would like to also mention it here. This is one of the projects we did. It was Phylogenomics. This is a monospecific tribe endemic to Guinea. This is also the only tribe that was not sampled in Darren's phylogeny published in 2022. He also mentioned this in his talk 2 years ago. So he was really wondering where this species will be placed in the phylogeny. In the past, some people said probably it would be Sonorili. And our collaborator, Pepe Haba, he recollected this species in 2 expeditions in 2017 and 2019. So this gave us an opportunity to uh, test the phylogenetic placement of this tribe. So we first, in the same way as we did for Medina mirabile, we used target enrichment data. Um, as we see here, it's well supported to be closely related with Asian dislocated and the Meranthema mirabile, but the coalescent phylogeny and the concatenation phylogeny are not really consistent. Based on the coalescent phylogeny, it's Meranthema, Meranthema mirabilis is sister to dislocated, but in the concatenation phylogeny, Phylogenomics is sister dislocated. So we wanted to really investigate this. Because when we were doing sequencing, we did a HyperSeq, so we also had genome skimming data, and we wanted to see if we can use skimming data to resolve the problem. After employing genome skimming data, we had more than 11,000 loci, and this is the resulting phylogeny. Like there's no discordance anymore. Now Meranthema is sister to dislocated, but we thought, okay, maybe we can still check the gene tree conflict. And indeed, this node presents the most gene tree conflict. And we also did plastic phylogeny and found that this is not consistent with the nuclear phylogeny. So we did phylogenetic networks to detect possible gene flow. These are 4 different scenarios. So network 0 means reticulation is not allowed. And then we increased the maximum number of reticulation allowed. So from 1 to 3, this is the statistically best model, and that supports the hypothesis that there was gene flow in the evolutionary history of meranthemum from dislocated, which result in the conflict we observed. Yeah, so in conclusion, within Old World Melastomataceae, we reinstated meranthemum and placed it in dislocated. We also reconstructed the phylogenies of African Melastomataceae, fleshy fruity sonorili, and the whole sonorili. We also placed Phylogenomics, and we also showed that by combining angiosperm 353 and Sanger data, we could cost effectively place taxa in the plant tree of life. We also showed that by using an inclusive reference, we could effectively integrate diverse NGS data. So that's all for the talk. Yeah, okay, I also need to thank my supervisors, Gudrun Kaderai and Mary Claire, also the whole AG Kaderite, also lab technicians and my great collaborators, also PhD committee members, Fabian and Dirk, also all the library that provided material. So that's it.
Thank you so much. I'm sure there will be quite a few questions. I have a few, but I will open it up to the floor first. Anybody, if you have a question, just unmute yourself and shoot. Go ahead.
Yeah, I can start with a question.
Go ahead, Leopold. Thank you.
Thanks, Luo, for your very great presentation. I have a question on the probe set, well, on the reference set you designed for the sonorili. So I guess you got a better resolution with this high amount of genes, right? It's kind of crazy, like thousands of genes.
Yeah.
But do you think we could apply your approach to have a reference set for the whole family instead of only sonorili?
I mean, for the current reference we have, I think we can definitely apply it to Melastomataceae as well, because Melastomataceae was used as our group. And this is what we have now. And I think with more transcriptomes or even genome assemblies available, it is possible to develop a reference for the whole family.
I will follow up a little bit on that. So in that, for all of those, do you need to have the deep genome skimming, or were you able to use the previously obtained regular data from regular genome skimming, or the leftover from the probes, the enrich target enrichment to achieve that?
Yeah, this is also something that I found out. So the previously sequenced data can actually be used. So if you see from here, HypeSeq, it means what we have. So what
Hmm.
we did, genome skimming plus target enrichment.
Hmm.
I mean, some data, probably the material was really bad. So you could see probably nothing got. But for the good ones, you can get almost 4,000 loci.
Okay.
It's mainly decided by the quality of the skimming data.
And your of those 2,500 or almost 4,000, what is the average? How much missing data you end up in the end? Because some of the issues we have had is that you can recover a lot of loci, but then if the number of taxa that don't have those loci varies a lot, especially with the astral, you're getting some weird relations because there are things that the sampling is so low for many loci. How is there missing data there?
Definitely, at least, especially for the angiosperm 353 data you see here, there were at most, I think, 1,000 loci, and some were only probably represented by 20, something like that. But from my experience with Astro, it's okay. I didn't concatenate all these loci because of the missing data. Because for Astro, it was basically summarizing
Mhm.
the single gene trees. So yeah, I thought it was robust.
And did you get any mitochondrial data with this, or is it all nuclear?
I didn't try that with the
Okay.
mitochondria data.
Was thanks for the wonderful talk. It was great to see how you integrated the different sequencing approaches. I think it's very promising also to see that we don't have to throw away all the old stuff we have necessarily. But what be your recommendation for going forward? Do you think there's a specific method that works best if we now want to add into existing phylogenies? Might that be different for different groups?
Are we talking about Melastomataceae or
Melastomataceae,
okay?
but for the whole family. You've looked at a couple
Okay.
of tribes. So like, do you have a recommendation in general for the family? Do you think it's very group specific?
Um, so for us, um, but yeah, but I don't know. Maybe in other groups, the genome size might be very large, and you cannot really get as many loci as you expect. So like for me, the first choice would be transcriptome. That would be ideal, but not very possible sometimes. Then I would probably go for deep genome skimming.
Hey, Leon, nice talk. I have many questions. Have a
Go ahead.
nice presentation. Some specifics, some not. For example, regarding this the deep genome skimming, I think looking at the data from the genome skimming that we have from Sonorelli, I see that it kind of varies within genera, the results, and sometimes across genera, I mean the
Yeah.
recovery of loci. And I was wondering if might be like, because you mentioned it right now that the size is low, maybe lower compared to other taxa. But I don't know if we don't actually know much about genome size, right? Within Melastomataceae or even Sonorelli. And I think that is one of the things to think about the deep genomics scheme, which I really would like think that is the promise in every group, but it might be hindered by the genome size sometimes. And I wanted to know, like, what what are your thoughts about this variation of recovery of genes, even within Medinilla or within Sonorelli as a whole?
Yeah, as I said before, because within Sonorelli, we have some genomes assembled already. So we know the genome size is the largest one is less than 500 megabases. And there was a benchmark paper testing the optimal sequencing depth for deep genome scheming, and they advocated that if you have more than 10 times sequencing depth, and then you should have enough loci at least for phylogenetic analysis. Yeah, but so like if I want to work on another group of melastomataceae, I would first try a few samples, maybe 6 GB or 10 GB, and see how many I can recover.
Yeah, I see. That's nice. I also have a comment, like as Fabian mentioned about doing a mitochondrial data set. I kind of assembled some a mitochondrial data set, and it's nice to see the same result that you recover with the blastome, at least for Felicidami, that it seems that it's inside the Socadi clay, which is super interesting. But what I found it interesting, because I was just trying also to run some snack analysis within and across the family. When I ran within Disocati, I didn't find any introgression as you obtained it in the Disocati related to Felicidami. So looking at that result made me think about the level of gene trees or loci that you have to recover this type of introgression as well. I don't know about your thoughts as well when you showed like many networks, which one? So all of them are kind of which one would be the best configuration you think related to the circadian Felicidami?
You mean the best explanation I have
Yeah.
for
Yeah, the best network at least, because some of them are different, the configuration. Yeah, this one. Like, for example, the network one would be with Felicitema, which is cool, but the remaining ones does not relate necessarily with Felicitemi, right?
Yeah. I mean, for all those with reticulation allowed, like from 1 to 3, Felicidemia is sister to Asian dislocated, right? Then there are different scenarios. So when it was network one extinct clade provided a genetic component to Felicidemia.
Yeah, I see. Yeah, that's the only one that directly is is related to this tax, right? So yeah, that's interesting.
Yeah. The thing is, morphologically, I would say meranthemum would be sister to dislocating. Yeah, they are more similar, but somehow this is not really consistent with the results here.
This is very cool. Based on that, what about the tribe level, what you would consider? Disocati and Felicitemi.
Yeah, disocati is tetramerous, so is meranthemum, but Felicitemi is pentamerous.
That's cool.
Yeah, I don't know.
That's difficult because crossing the morphological. I don't know, I think you can use the networks with some morphology data, probably.
Yeah, the thing is, I cannot see the morphology of the extinct clade, so I cannot really like
Yeah,
compare.
that's true. Anyway, but that's cool. Cool work. And other thing unrelated, I don't know if your presentation for your dissertation will be available for those online, but I would
Yeah,
totally would like
it
to
will be
see.
hybrid. Yeah,
Oh, cool. If you want to share with.
I can send you the link.
Nice. Awesome. Awesome results.
Thanks.
Congrats, Luo.
Thank you.
Darren seems to have a strong opinion. He put here on the chat. Long live Phylogenomics.
Yeah.
I can see why.
Mhm.
So, I mean that this to me opens up another issue. I'm dealing with a different group where if which is a little bit of what you saw on your paper on um, uh, the fleshy fruit is that some data might suggest one taxonomy and other data or other analysis with the same data might suggest another way of doing taxonomy. And I think this is something we're going to have to grapple more and more is like it might be difficult to have a taxonomy that is perfect for all methods and all data unless we have this ginormous non-informative genera or tribes or families or whatever. So this is more a philosophical thing. Um, any other comments, questions?
I might have a very far-fetched question. I mean, I'm always thinking about flowers, but one thing that struck me when in your talk was that you said that Sonorili is the second largest tribe in Melastomataceae.
Yeah.
So, like just from the flowers, they seem quite different from Meconiae. And then they have 40% fleshy fruits. I guess Meconiae has more. Do you see parallels, like they might be ecological or evolutionary between the two groups, where you could see why these two tribes became so species rich? Or do you think very different mechanisms led to the species richness in Sonorili?
Mm.
It's very speculative, but I'd still be curious on your thoughts.
I would say it could be parallel evolution. Within the for the flesh fruit sonorili, Medinilla is apparently the most species rich one. So yeah, I would think it would be interesting to compare Medinilla and Meconia, or at least one clade of Meconia, to see the evolution of fleshy fruit, and also include the comparison of maybe animals.
Yeah, like at least in terms of flowers, the Medinillas seem to be way more exciting than many of the Meconias in terms of like how showy they are, how big they are. So they invest in both flowers and showy fruits, and Meconia invests apparently more in showy fruits. So that's, I don't know, that always recently that bugs me a lot.
Yeah, the fruits of
Of course,
Medinilla are
Juan.
much diverse
Yeah.
compared to the inflorescences.
I see your point, Agnes, but I think first it depends what we call exciting. In Melastom, in Mycone, the difference between the smallest one and the biggest one is more than 20-fold. So there is a big variation there in that sense, and then difference in colors and stuff. Not in the morphology of the anthers, but there is a lot of difference. But then if you go to within Sonorili, there's a huge of variation within the flowers of the capsular fruited ones. But once you get to Medinilla itself, there's variation, but I don't think it will be like I see it in the same order as as Myconia. So I don't know if the flesh, Ricardo Kribel has always talked about this, how maybe you're investing in dispersal or in pollination. Um, I don't know. Which also affects taxonomy in a way, because then we end up subdividing things that have capsular fruits and then lumping things for the most part that have fleshy fruits. So. Any other thoughts? See, Juan thinks that myconia flowers are cool. So, but there is
I didn't mean to
inside
say they're
bias.
not cool. I was very, very puzzled by all the different scents that Meconia flowers produce recently.
Yeah.
So
Yeah. So, yeah. So if there's no other questions, we'll have another. We have already booked our next talk for the end of March, the last week in March or second to last week in March. Can't remember right now. So the announcements will be going out. And again, an invitation to any and all of you to present either finished projects, halfway projects, or even just halfway baked ideas of things you want to talk about. It will be great to hear more. And everything goes: anatomy, pollination, systematics. We're all happy. And again, Luo, thank you for a great talk. And I look forward to the actual dissertation next week.
Thanks. Yeah.